Espandiol summary. Pantothenic acid, also known as vitamin B5, is a water-soluble vitamin that is a precursor in the synthesis of coenzyme A. Coenzyme A is essential to many biochemical reactions that sustain life. Also, the phosphopantothenial moiety of coenzyme A is required for the biological activity of several proteins, including the acyl carrier protein involved in fatty acid synthesis. More information. Pantothenic acid is essential to all forms of life. It is ubiquitously found in foods of plant and animal origin, and dietary deficiency is very rare. More information. The Food and Nutrition Board of the U.S. Institute of Medicine set an adequate intake (AI) of 5 mg/mg per day for adults based on the estimated daily average intake of pantothenic acid. More information. Evidence from limited intervention studies suggests that pantothenic acid and/or pantothenol alcohol analog might improve the healing process of skin wounds. Yet, additional larger studies are warranted. More information. Treatment with high-dose pantothene, a pantothenic acid derivative, has been shown to lower serum cholesterol and lipid concentrations. Although pantothene therapy appears to be well tolerated, medical supervision is indispensable. More information. Foods rich in pantothenic acid include animal organs, liver and kidney, fish, shellfish, milk products, eggs, avocados, legumes, mushrooms, and sweet potatoes. More information. Little or no toxicity has been associated with dietary and supplemental pantothenic acid such that no tolerable upper intake level (UL) has been set. More information. Pantothenic acid, also known as vitamin B5, is essential to all forms of life. 1. Pantothenic acid is found throughout all branches of life in the form of coenzyme A, a vital coenzyme in numerous chemical reactions. 2. Function. Synthesis of pantothenic acid cofactors. Coenzyme A. Pantothenic acid is a precursor in the biosynthesis of coenzyme A, CoA, Figure 1, an essential coenzyme in a variety of biochemical reactions that sustain life. See below. Pantothenic acid kinase 2, PANKII, catalyzes the initial step of phosphorylation of pantothenic acid to 4-phosphopantothenic acid. Coenzyme A and its derivatives inhibit the synthesis of 4-phosphopantothenic acid, but the inhibition can be reversed by carnitine, required for the transport of fatty acids into the mitochondria. 3. The subsequent reactions in this biosynthetic pathway include the synthesis of the intermediate 4-phosphopantothane, as well as the recycling of coenzyme A to 4-phosphopantothane. Figure 1. 4-phosphopantothane. The 4-phosphopantothenial moiety of coenzyme A can be transferred to enzymes in which 4-phosphopantothane is an essential cofactor for their biological activities. See 4-phosphopantothynylation. Cofactor and co-substrate function. Coenzyme A reacts with acyl groups, giving rise to thioester derivatives, such as acetyl-CoA, succinyl-CoA, malonyl-CoA, and 3-hydroxy-3-methylgluteryl, HMG-CoA. Coenzyme A and its acyl derivatives are required for reactions that generate energy from the degradation of dietary fat, carbohydrates, and proteins. In addition, coenzyme A in the form of acetyl-CoA and succinyl-CoA is involved in the citric acid cycle, in the synthesis of essential fats, cholesterol, steroid hormones, vitamins A and D, the neurotransmitter acetylcholine, and in the fatty acid beta oxidation pathway. Coenzyme A derivatives are also required for the synthesis of the hormone, melatonin, and for a component of hemoglobin called heme. Further, metabolism of a number of drugs and toxins by the liver requires coenzyme A. 4. Coenzyme A was named for its role in acetylation reactions. Most acetylated proteins in the body have been modified by the addition of an acetate group that was donated by the coenzyme A thioester derivative, acetyl-CoA. Protein acetylation alters the overall charge of proteins, modifying their three-dimensional structure and, potentially, their function. For example, acetylation is a mechanism that regulates the activity of peptide hormones, including those produced by the pituitary gland. 5. Also, protein acetylation, like other post-translational modifications, has been shown to regulate the subcellular localization, the function, and the half-life of many signaling molecules, transcription factors, and enzymes. Notably, the acetylation of histones plays a role in the regulation of gene expression by facilitating transcription, i.e., mRNA synthesis, while deacetylated histones are usually associated with chromatin compaction and gene silencing. The acetylation of histones was found to result in structural changes of the chromatin, which affect both DNA-protein and protein-protein interactions. Crosstalk between acetylation marks and other post-transcriptional modifications of the histones also facilitate the recruitment of transcriptional regulators to the promoter of genes that are subsequently transcribed, reviewed in 6. Finally, a number of signaling molecules are modified by the attachment of long-chain fatty acids donated by coenzyme A. These modifications are known as protein acylation and have central roles in cell signaling pathways. 4. Phosphopantotheniation-specific multi-enzyme complexes, which need to carry out several reactions in an orderly manner, may require the covalent attachment of a 4-phosphopantothene arm to a carrier domain, or protein. This carrier domain